Hi everyone, Dr. Bruce here. This week's video is going to be on a sticking point in muscle physiology, which is the physiology of muscle contraction. I know this is a complex process, and a lot of students have trouble with this. So I'm going to go through this. So I'm on my website here, and I have a page where I developed a lot of interactive learning activities. You'll see some of these in your course. I'm now working on 3D models, so that's been a big project I've been working on recently to try to get 3D models onto my website. So let's take a look at this muscle contraction physiology interactive activity, and I'll give you the link to this in the announcements below this video. So let's take a look at this. All right. So first, this activity begins with, like a skeletal muscle. Like, what is skeletal muscle? It consists of bundles of fibers, and there are various parts to that. So really, it's bundles within bundles, with the smallest unit being the muscle fiber. Sometimes that's called the muscle cell. So that's what muscle consists of. And you might remember from tissues that skeletal muscle is striated. You can see the stripes there, the striations. And if we enlarge that, what we see here is a contractile unit called a sarcomere. So between those striations, we have this sarcomere. And the sarcomere consists of two contractile protein filaments, which are called actin and myosin. So, and you can discover that by clicking on these blue boxes. So, actin is like a double helix protein filament, sometimes called the thin filament. It's on the outside. On the inside is myosin, which is a thick filament with globular protein heads. So these heads are going to form, they're going to connect to actin and form what are called cross bridges. All right, so if we look at the structure of this contractile unit here, the sarcomere, you can see there are some different lines and things that you can learn. But we're going to move on to looking at actin and myosin in a little more detail. So actin, remember, a double helix protein, so two strands are wrapped around each other, but then there's another protein complex wrapping around the actin called the troponin-tropomyosin complex, and that's this thing. So the green part here is the troponin, the purple is the tropomyosin, and it wraps around the actin and covers these little blue things, which are called the myosin binding sites. Myosin is a little bit different. It's like bundled proteins that have these globular protein heads. These are going to connect to the myosin binding sites on the actin. So the process begins where a motor neuron connects to a skeletal muscle, which is called the neuromuscular junction. So the motor neuron, this is myelinated. So these are this is myelin, and myelin is a lot faster than unmyelinated axons. That's going to carry the electrical impulse, the action potential to the muscle at the neuromuscular junction. If we were to magnify that neuromuscular junction there, we would see on the muscle side, we have an area called the motor end plate. So it's enfolded. It's got little ridges and folds in it. And there are some transport proteins in there as well. So we can, click on these blue boxes and see what all these parts are. So here's our motor neuron again. Here's the axon terminal. So the action potential comes down a motor neuron and hits the axon terminal, and then it causes these calcium gates to open. So there are these calcium channels. Calcium moves in. So these are voltage gated. They respond to the action potential. So calcium moves in and then causes release of the neurotransmitter, which is acetylcholine. So in skeletal muscles, the neurotransmitter is acetylcholine. Acetylcholine then moves across the synaptic cleft to the motor end plate, which is on the muscle. Acetylcholine then connects to receptors on the motor end plate and causes sodium channels to open. So sodium comes in because muscle is also polarized, and we're going to get some depolarization there. All right, so this is a little animation I put together that goes through the whole process. So there's our action potential. There's the calcium coming in. It attaches to the synaptic vesicle, causes the release of acetylcholine, hits the motor end plate. Sodium then goes in, opens up the sodium channels Sodium goes in and the motor end plate depolarizes. This is kind of the same thing that happens in neurons, so it's a very similar process. So what happens next? So before I can explain what happens next, we have to talk about another structure called the sarcoplasmic reticulum. So what is this? This is a network of membranous channels that wraps around the outside of the muscle fiber. So it's on the outside. So it's a system of tubes, these channels. And so, like what does it do? Like why is it there? Well, what it does, you're going to find out that you need calcium in order for muscles to contract. So where does the calcium come from? Well, it comes from the blood. Well, where does it come from in the blood? It comes from the calcium you eat, right, in your diet. And if you don't eat calcium, it comes out of your bones. So the osteoclasts take that calcium out of the bones. So it's in your blood, and then the sarcoplasmic reticulum captures it from the blood and stores it until it's needed for muscle contraction. So that's all that does. So here's like an enlarged portion using my fancy PowerPoint, images. And so here's like a tube of the sarcoplasmic reticulum, and you'll see there are two types of channels here. So the two channels are both calcium channels. One is an active transport protein, the other one is voltage gated. So how does this work? Well, what's happening, like I said, there's calcium in the blood, and the muscles need the calcium in order to contract. So in order to pull that calcium out of the blood, we need a transport protein that uses ATP. So we create a calcium gradient inside of these tubules. So this transport protein is pulling calcium from the blood and sort of stuffing it in there, creating a big calcium gradient so that we can use it for muscle contraction. The other transport protein is voltage gated, so it responds to depolarization. And depolarization happens when the muscle is ready to contract. So the nervous system, sends the impulse via the motor neuron, you get the acetylcholine, opens up the sodium gates, and the motor end plate sodium flows in, depolarization. Depolarization spreads to the sarcoplasmic reticulum, opens up these voltage-gated calcium channels. Now this thing is stuffed with calcium, so calcium is released into the muscle cell. Okay? And that's what this animation shows. So, at rest, the calcium is moving into the tubule via the active transport protein. But then once the depolarization happens, the calcium now moves out of the tubule and into the muscle cell. So what does calcium do? Well, calcium attaches to the troponin part of the troponin-tropomyosin complex. And so I created this little animation. This animation took so long to create because I'm not an animator, but it was just really took a long time. Anyway, calcium attaches to the troponin on the troponin-tropomyosin complex, causing the whole complex to move and exposes the myosin binding site on the actin. So calcium attaches, the whole thing moves, myosin binding site is exposed. Right, so here's another animation that took a long time. That just a few seconds took like hours to get right. So what happens when the myosin binding site is exposed? Well, myosin binds to actin, and now it can push or pull actin along. Well, that's going to require energy, right? So at the myosin head, there's an enzyme that will hydrolyze ATP and use the energy from ATP to power that movement. That's called a power stroke. So ATP attaches to the myosin head, pulls the actin along, and what happens is we get shortening of the sarcomere and muscle contraction, and that's it. So that's really the whole story. So the sarcomere shortens, and since the muscle consists of tons of sarcomeres, next to each other, the entire muscle contracts. All right. So that's really the whole thing. So what I'll do is I will put a link in the announcement for this to go to this activity if you want to work through this activity, and hopefully this will help you understand muscle contraction a little bit better. If not, you can always ask questions. I'm available via the inbox or my email, and I'm happy to help in any way I can, and hope you're doing well, and we'll see you in cyberspace.